absolutely. Hello, and welcome to Microbiology. This morning I'm going to be talking about specific viruses and a little bit about extra content about the coronavirus. Now, as part of our class here, we looked at viruses kind of early in the year, looked at viruses and vaccines. Today we'll probably just take a look over viruses. I might mention vaccines a little, but vaccines are probably something I'm going to come back to once we're kind of finished with the core virus content. It's likely you will get an exam question either on the biology paper or on the microbiology paper about viruses, or given the year that's in it, it's possible you'll have both. Now, just to talk a little bit about viruses themselves. Viruses aren't alive. Well, they're technically not alive. If you remember from the characteristics of life we covered, we mentioned growth, reproduction, response, directed movement, all that funky stuff for living things, and viruses don't really do any of that. What it does mean is that they're kind of alive, in that they're alive when they get access to other living things. Now, what does that mean? Well, hold on a second, I'll just do me. Right. So, they don't have any cell machinery of themselves. They have to co-opt the cell machinery of another living thing. So without a host, viruses aren't alive at all. When they're inside a host, they're kind of arguably alive because they take over the virus or the host cell's production. So if you look there, there's a generic sort of virus model on the screen. They have a core of nucleic acid, DNA or RNA, surrounded by a protein coat. Now, DNA and RNA are very fragile. They don't last jig time outside of that coat, which is important, and we'll talk about that later. You'll also notice that on the outside of the virus, they have a lipid coat. Now, they steal that lipid coat from host cells, but it means that their exterior is made up of fat molecules. So if you want to get rid of viruses on a surface, get rid of the fat molecules, you get rid of the viruses, because you break down the lipid coat, exposing the DNA, and if you expose the DNA, it'll break down very, 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 very quickly. Also, critically, you can see there that there are protein spikes. Now, those protein spikes are known as antigens, and they are a way that viruses hook themselves on to cells, infiltrate into cells, but they're also how your immune system tags them. Those antigens are incredibly important. We'll be coming back to talk about them more later, about how you fight viruses. They also have some I don't have a protein coat inside them, but again, that sometimes they have a lipid coat, sometimes they have a protein coat. They usually have a lipid coat, and sometimes they have a protein coat. But if you can break that down, you expose the DNA, that's it pretty much. They're kind of that point. And they're not a lot beyond that. They're RNA and a stolen protein coat and some protein spikes and a stolen fat coat. And that's kind of it. They're quite simple and basic. But that RNA or DNA is very important because that allows them to co-opt and take over living processes or proper living cells. So, once inside a living cell, viruses can replicate with the host cell, and that means they're definitely alive. So they have the features of both living and non-living material as a result. They can multiply inside living cells, and they're called obligate parasites. That means that without a host, viruses die. And unlike bacteria or fungi, they cannot live for very long without a host. Bacteria and fungi can both kind of basically turn themselves into spores or endospore up and basically become resistant to the outside environment and wait for a long time. Days, weeks, months, longer in some cases. Viruses really can't. Viruses have a limited shelf life outside of the body. The longest a virus has been observed living outside a host is 28 days. And that's pretty exceptional. Most of the time you're talking 24 hours, 72 hours, maybe a little bit longer. Because they are parasites, they tend to affect us and any of their hosts negatively. Why? Well, they turn our cells into virus factories. And, you know, we don't want that, because all of a sudden our cells, rather than doing what they're meant to be doing, I mean, muscle cells or skin cells or whatever, they're little virus factories. And all those viruses go out and they infect cells around them. So usually you feel terrible when you've got a virus, because millions of your cells have been turned into these virus factories, and they're vomiting forth more viruses, and they're infecting more cells and more cells and more cells, until one of two things happens. Your immune system gets a handle of it, or potentially you die. Now, there are other outcomes, but those are the two most common. Viruses require a host. That means that if there's no humans around, there can be no human-borne viruses. There can be animal-borne viruses if there's animals around, but there needs to be living hosts there. They can't just move around on their own, which is a good thing. It means that it limits where they can go. They cannot, if we wanted to cultivate viruses for study, we have some challenges. Because they can only multiply inside living cells, they're called obligate parasites. If I want to cultivate in a lab bacteria or fungi, I can just pop out agar plates and I can inoculate onto them, and I can cultivate my viruses, my bacteria or fungi there. I can take a sample, I can isolate it, I can study it, I can work with it. It's fairly straightforward. Because viruses don't have that you can't cultivate them like that. It means that you need to effectively have a living host for them. And that requires additional ethical controls. And it also means in some ways these things are a little more dangerous, because you'll be working with a living animal or a living host in some way. That's ethically more complex, but it also is more fiddly, because you're working with a monkey you've infected with a virus. The monkey scrapes you, you get infected with the virus. And you're more like, bacteria's never going to jump off the plate and go for you. But a monkey you've infected with one potentially can do. So, living studies are ethically complex for that reason. Because they lack host cell machinery, the crown jewels in our medical arsenal, the antibiotics, have no effect against viruses. I could shoot you up with as much antibiotics as you want. It'll, at best, have no effect against the virus, and at worst, make you a little more susceptible to the virus by suppressing other aspects of your immune system. So, antibiotics are of no use here. And that's one of the reasons why viruses can be so scary, because we don't have one big gun we can go to immediately and fire back at them. We don't have that. So we have to use different tactics instead to defend ourselves. Now, one of the things about viruses was there was a theory for a while in science that viruses were alien invaders to a certain extent. And that they'd come from outside our biosphere because they were so different. They didn't have any of the kind of characteristics we associated with living things. They were these little bits of something that co-opted cells, and we weren't sure how they worked. And they were so small. If you look there, that's basically 1 micrometer, which is 1,000th of a millimeter, and a millimeter is about the width of your little finger or little fingernail. They're very small. So the first time we actually see them, we only get an electron microscope. And the first virus we saw was a bacteriophage. And so there was still kind of the argument of, were they alien invaders, were they from somewhere else? And then they saw the bacteriophage and everyone freaked out. Because if you look at the bacteriophage there, it looks a little like an alien lander. So everyone freaked out for a bit. Now, they're not, as far as we can tell, like some panspermia thing where they landed here from a different planet. They evolved here, but they looked really weird and their behavior was a bit strange. They're very different from the bacterial and fungal roots of life. So they came along. We're not sure from where. We don't think it's from space. We don't think they're alien invaders. But the first time people saw a bacteriophage, everybody freaked out. They're really small. You don't kind of quantify how small they are, because you think, 1,000th of a millimeter, that's pretty small. That's really very, very small. You'll only see them with an electron microscope, which complicates scanning for them. If you want to find viruses, you can't look for them. I can't, like if you have a bacterial infection, I can stick it under a microscope. I can take a look at it under a microscope, and I can see it. You can't do that with things. So we've got to look for other signs of a viral infection. You can't just, you won't see it easily, certainly. Now, let's talk a little bit about what the coronavirus looks like. The coronavirus is so called because the antigen spikes on it form a little bit of a crown. You see the way they kind of hang out there? There's kind of 3-lobed one there. They form a little bit like a solar corona. So they're called that. They're called the coronavirus for that name. And a coronavirus it's quite a common type of virus. The official name of the one that's currently troubling us is SARS-CoV-2, and the disease that it causes is COVID-19. It's so called because it's a coronavirus that developed in the year 2019. It was first described in China in December 2019 around the city of Wuhan. Now, how do viruses replicate? Well, we've mentioned before that they turn your cells into virus factories. Now, we have a diagram here for a bacteriophage, but this works broadly similarly on every virus. The bacteriophage bops along here. It attaches itself to the virus or the bacteria it wants to affect now. This can also be as easily a coronavirus infiltrating a human cell. Same principle applies. It hooks itself on now. It uses its antigens typically to hook on. And then the virus inserts its DNA into the host cell. Now, in addition to its DNA or RNA, it has some viral enzymes which break down your DNA. So it'll get into your nucleus, it'll break down your DNA. And then your cell is kind of rudderless. Normally, your nucleus tells your cell what to do. Do this, do that, make that, make that. It doesn't have that option now. What it does have is a bunch of viral DNA, which is going to take over what your cell does. So what does the virus want to do? The virus wants to propagate and survive. How does it do that? It goes into your cell machinery and it turns it into a little manufactory for more viruses. So your cell basically turns into a little virus factory. And it builds, basically puts together the DNA, it puts together any virus enzymes you need, any protein coat you need, and then out it pops through the side of the cell, stealing your lipid membrane as it goes. Now, the one sign of that happening is that on the outside of the host cell, those little antigens are being made and they're being pushed up and they pop on the outside of that cell. And that's a warning sign to the body that that cell is infected with a virus. And then viruses just either, the whole thing pops up and then basically the whole thing pops up and out, or there's this continuous flow, which is referred to as viral load, where it keeps coming out. The diameter of a cell versus the 1000 millimeters of the COVID virus. You're talking about most viruses are going to be probably, for humans, human cells vary in size hugely, but you're probably talking about, it's going to vary from, in some cases, kind of maybe 1/10 of a millimeter down to so like 100 micrometers down to about 20 micrometers. So a virus would usually be a fraction the size of a cell, like very small in comparison. That diagram is purely kind of notional sort of description there. So what happens is, though, the virus starts making them. Now, it's either the whole thing fills up and it bursts like a balloon and viruses go flying out and infect everything, and you'll feel terrible when that happens. When that replication cycle happens, that's why people feel so sick when they've got a virus. Their body is like, oh my god, whew, my cells are all dying, and they are all dying. Well, they're not all dying, but a bunch of them are dying. That's why you feel. That's why you feel terrible. Or it's continually popping out and infecting more. And rather than those cells doing what they're meant to do, they're suddenly viral factories, spewing out more viruses all over the place. All right. Now, what do viruses do in humans? Well, common ones that we have to worry about are measles, mumps, and rubella, and the cold and warts. Now, if you were to look at the infection rates of viruses, COVID-19, for example, or SARS-CoV-2, it's, quite a high infection rate. It's nothing on measles. Measles is a far more infectious, rapidly spreading virus. But in Ireland, you will notice that most people are vaccinated against measles, mumps, and rubella. Not everybody, but most people are vaccinated against them. And that means that there are very few hosts to carry that virus, which is great. More common ones like the cold, which mutates so much we don't have an effective vaccine against, and mostly makes people a little sick. And the warts, which again, is highly infectious, but is relatively easy to treat. It's not a big concern for us. Plant diseases like mosaic disease make stripes in plants, and tobacco mosaic virus, other ones they do that. Dutch elm disease, a bunch of other ones like that. They're very bad for plants, and plants don't have the same kind of immune system we do. Are you going to die? Well, eventually. Do you mean more Kenzu? Would you mean the more a shorter time frame? Eventually, yes, but not unlikely from this. But we'll talk a little bit about coronavirus about this. Now, animal viral diseases, rabies, very nasty, but quite reasonably well contained, mainly transmitted through infected bites from animals. Rabid animals are mostly killed to avoid this. Foot and mouth in Ireland, we had a big issue around the early 2000s, and we shut down the country to avoid that one spreading too. And then the SARS. Now, SARS is a very closely related to coronavirus. SARS had a much higher lethality rate. SARS had about a 10% mortality rate, which is very, very high, much higher than COVID-19. But SARS didn't spread nearly as well, and the symptoms showed up a lot earlier. So you didn't have the same problems. Basically, COVID-19 is a different problem. But SARS taught a lot of East Asian countries about effective ways to contain these diseases. And so they had facilities kind of ready to go, which is one of the reasons why China and South Korea kind of handled this quite well, is because they had experience. SARS had been a training wheel for them, so they learned how to counter a lot of the problems here. Now, is there any useful stuff? Are the viruses just a disadvantage like mosquitoes? No. We use viruses a lot nowadays. So, for example, we can use viruses to insert different types of DNA into cells. For example, in humans, you can use retroviral treatment if their human is missing a DNA strand, they can't make a certain type of material. In some cases, you can break down and insert the healthy gene into cells, and it inserts it into the genome, and then that person's cells that you've infected are kind of fixed. Now, that's very difficult to do for a whole human. Like if someone has type 1 diabetes and they can't make insulin and they're insensitive to insulin, putting in the missing DNA strands theoretically would fix the problem, but infecting a whole person with the virus like that is very difficult, specifically because these viruses are designed not to turn the cells into virus factories. It's just to insert their DNA and move on. This is also how we engineer bacteria to make certain drugs. So if you want to make a drug, you can make a virus that instead of coding for viral components, codes for the drug. It inserts that into a bacteria, and then that bacteria just makes that drug. And you make 1 billion of those bacteria in a big bioreactor. You decant them off, you split them up in centrifuges, you separate out the drug, you use chromatography to separate down to its pure form, and bam, there's your drug. Like if you want to look at, we used to kill an awful lot of dogs making insulin, and then we killed an awful lot of cows making insulin, and everyone was getting very upset about that. So we found a way to do it with bacteria, and no one gives a toss about bacteria. So we were able to suddenly manufacture these things in large numbers, which is great. So those are the advantages. Now, where does SARS-CoV-2 come from? It's believed to originate in bats. The bat immune system is highly alert. So the virus needs to be very stealthy or grow very fast to evade the bat's kind of metabolism and immune system. They have a very quick metabolism. They're about 4 times faster than a mouse. So if you are a virus and you have evolved to deal with the bat immune system, getting into a human is like suddenly you're fighting someone in slow motion. In comparison to a bat, they're all over you. The bat immune system is all over you really, really quick. But it's unlikely for the virus to go straight from bat to human. They usually go through an intermediate host. We believe in the case of SARS-CoV-2, it was the pangolin, which is a type of spiny anteater, which are, in many ways a lovely little animal who walk around looking like they need to apologize for something terrible that they've done, and in this case, is they're kind of apologizing for their error. One of the beliefs is that pangolins got infected by the viruses, and there either may have been pangolins in the wet markets in China, or it's possible that there's a bunch of different places, but they think it traveled through pangolins to get to humans, because often a virus can't go directly from one animal to another. There's often an intermediate species in the middle that the virus has to travel through. So, for example, you usually don't get avian flus from birds. It often gets through an intermediate species like a pig, which then infects humans. So the virus can't hop along just immediately. It's got to hop along somewhat slowly. Did Batman choose bats to survive SARS? He might have chosen bats because, some people are freaked out by bats more, or bats' aggressive immune systems, or their very fast metabolisms, all of which would be an advantage for a crime fighter. Also, criminals cowardly, superstitious lot. Batman is really at the moment better off punching billionaires, I think, than punching poor people. So, one would hope, if I was writing Batman, it would be a lot more. And then, Batman has to take down, this evil corporation, or that dubious ethical practice, or, at least if he's, beating up low-level criminals, it should probably be to liberate supplies of hand sanitizer. Now, what have they found out about SARS so far? Well, the primary thinking is it's droplet dispersed, which means that if someone coughs in the air, it's the droplets of moisture that are going to infect you. It's not going to kind of drift around like some kind of terrible death fog infecting people. They think that at most it's viable kind of in that sort of aerosol form for about 3 hours, which is long enough. Now that 3 hours is at the far limit. They found that on plastic and steel, some of it would survive up to 72 hours. Now, that doesn't mean, oh my God, someone touched this table 3 days ago and they had SARS CoV-2, so I'm going to get COVID-19. Not necessarily, but it does mean to kind of clean those hard surfaces like that. Cardboard, it was lasting up to 24 hours. Copper, up to 4 hours. The most likely person you are to catch it off is an infected human. By far. If you're not around humans at all, you're good. Now, not being around humans at all is difficult, particularly if you have to work, you have to commute, you have to get to places. But which is the reason behind the kind of social distancing and quarantining. If you can avoid humans, you can avoid getting this, Like if you're some nutter living in a farm in the middle of nowhere and all your food is prepared and you've got electricity generators and everything, you'll see this out, no problem. The rest of us are, have got, dangers from being around people. Hence, the social distancing is the primary strategy. There are, the Netherlands and the UK have proposed a different strategy where people try to acquire herd immunity. I'm deeply skeptical of that as a plan, because of the initial body count you'll get out of doing it, and like the plan only works if it doesn't mutate, and if it mutates, I guess we start over, which is which will not be the best. Characteristic signs and symptoms of it. Patients with COVID-19 usually have mild to severe respiratory illness. The symptoms include primarily fever, a cough. Now, it's a dry cough, not a phlegmy cough, and shortness of breath. Now, fever, dry cough are the two big ones. Shortness of breath, people definitely observe. And you can get symptoms up to 2 to 14 days after exposure. Now, most of the time, there seems to be a 2 to 5 day period where you're asymptomatic. You don't have any symptoms, you feel fine. You've no cough, you know anything like that. But you're infectious. And this is how it's been so good at spreading. Has anyone ever played the computer game or app game Plague Inc.? The best way to start with a disease is to have a long asymptomatic period, because that allows us to spread it all over the shop before people realize it's a problem. And that 2 to 5 days is the killer, because if you go, people were talking, well, why aren't they stopping people coming out of planes? The difficulty with that is there's no sign at the moment. Until recently, all our ideas are, well, we could take their temperature, sure, but the problem with taking the temperature is, for 2 to 5 days they're normal, but they're still infectious. You will often then be sick for about 2 to 14 days after that. So you'll have 2 to 5 days of being asymptomatic but infectious, 2 to 14 days of symptomatic but infectious, and then you can remain infectious for more time after that. There's some cases they're looking at people kind of after initial infection, 28 days later, they were still infectious. So this is a disease that hangs around for a while, which is one of the reasons why the current situation, we could easily be looking at a month of a lockdown to try and get it initially under control, and then we may open up again. What's going to be interesting is China had been shut down since January. They're starting to open back up now. They've got limited stuff going back on. They're starting to warm their country back up and get it running again. What'll be very interesting to see is, does this all start back up again, or have they controlled it? They've been very aggressive in what they've done in some things that we probably wouldn't be willing to do, but they did a lot of other things that were very organized. For example, in China, if you go into any building, more or less at the moment, any public building, they take your temperature. If you are running a fever, you will be sent to a fever clinic. In the fever clinic, they will be a medic will come out to you in full protective gear. The medic will examine you, will take the temperatures, ask you your symptoms. Then the medic will send you in for a CAT scan. And what they will be looking for in the CAT scan is they will be looking for these kind of, they're called ground glass bodies in the lungs. These kind of partial, they're not exactly obstructions, but they're little, like kind of masses in the lungs, which are the kind of nearly diagnostic signs for this. Not exactly, but nearly diagnostic signs for this. In Ireland, getting a CAT scan like that is a big deal. It is, you can be waiting yonks to get one. They had portable machines, small machines they were using, and they were cracking through about 200 people a day there. So they'd scan you, and they give you your scan, they examine it, and if there was a mass like that on it, that doesn't necessarily mean, could be pneumonia, could be something else. They will go, okay. They will bring you in, and they will take a swab from your nose, and the way they get a swab is they get basically like a swab, you've ever seen, it's like a really long cotton bud, and they shove it right up your nose, which is unpleasant. They take it out, and they send that down to the lab to be basically cultured with, effectively not like, it's like an antibody screen, which is selective for selective for the disease. That will take about 4 hours for that to get back to you. There's an Irish crew who apparently have gotten a test that works in 15 minutes, which is great if they can get that reproduced well. So they'll do that test, and you'll sit then in a lobby, and you'll be basically staying kind of 2 meters away from everybody else, because everyone in that lobby is potentially infected, so everyone's a bit paranoid about, oh, maybe I'm just a bit ill, whatever. They'll stick you in that lobby for 4 hours. They get the results back. If they're positive, you will be you were brought along to a big facility they built, and this facility is, they look a bit like camps, but what they are is, there's a bed, there's kind of separate space given to you, and they've got nurses and doctors in full protective gear walking around keeping an eye on everybody. And you get into you have the bed there. They get you up for exercise periodically, a little bit of dancing, light exercise, stuff like that. The goal is that to help identify the people who are genuinely very, very sick that way. And the other reason, if you've got regular pneumonia, it can help knock loose that. So if you're up and active, you can help dislodge that. And primarily because older people get this most severely, it's a good idea to kind of keep them moving and see, okay, what's the problem? Is this just regular pneumonia, or is this pneumonia symptoms caused by coronavirus? So that's the goal with that one. We in Ireland, the most exciting thing we've kind of done beyond the aggressive measures taken to distance everybody is we're putting together a drive-through centre in Croke Park, much like the ones that were put together in South Korea, which have been very effective at screening. To finally get rid of this virus, after that, we need an extensive testing regimen to track who has it. Because even if people are isolating inside their homes, it means that if one member of the family is going to get it, the rest of the family are probably going to get it because you're asymptomatic for so long. So they knew that to fight it out, they had to get it down to identifying individual families and quarantining them. That's where, if we want to beat it, we're going to end up going. And it's been fairly positive in Ireland so far in terms of what they developed. So that's a good sign. Now, how serious is it? You'll hear a lot of people saying, oh, only old people get this. No. The current best knowledge is that those who get COVID-19 break down into 3 groups. Or after you've gotten it, there's kind of 3 groups you fall into. 80% of people get what's called a mild case. Now, a mild case can mean you have a cold. Or it feels like a cold. Fever, dry cough, feel a bit rotten for a few days, then you recover. It can also mean you need some medical attention, not including oxygen. That could be antipyretics, that could be a number of other things. There's been a bit of misinformation flying around about you're not meant to take ibuprofen. At the moment, that's not definite yet. But paracetamol is often more recommended because it's an antipyretic, it reduces temperature. Why are obese people more likely to get it? It's a higher risk factor, basically. We're not sure why, but you're more likely to get seriously ill if you're obese. In Seattle, where they were having to do heavy triage, if you were going to go, had criteria for going on a ventilator, and you had to be under 40, you had to have no prior major health issues with regards to your lungs, and you couldn't be obese because they were that short in ventilators. So they were making the choice based on the people most likely to survive. This is at the 5% we'll get to in a second. So 80% of people are mildly ill, but mildly ill can still mean really sick at home. Could mean 2 weeks of sweating like mad, being very, very ill. 15% of people will require some medical intervention, up to and including oxygen mask to assist breathing. So those people will need to go to hospital because they will need breathing to assist them because they're going to struggle to breathe. 5% of people will require major medical interventions such as ventilators or ECMO. Ventilators means that your lungs literally aren't taking the oxygen in the air and you need, so you need a pump to force them into the lungs because they're too filled with fluid. ECMO is even more serious. It's a major medical intervention where your lungs are completely not working, at which point what they do is they directly oxygenate your blood by inserting a line into your arm, and they effectively have like a portable lung. We do not have a lot of ECMO machines. They're a dangerous and drastic intervention to use because the risk of a bacterial infection from that is serious. If you are older, if you're obese, if you smoke, they make you more likely to be in any of these categories. Now, the World Health Organization, the initial mortality rate they thought for this was about 1%, 2%. The World Health Organization in China put it at 3.4. If you looked at Italy today, the mortality rate's at about 8%. Now, that's very high. We think it's higher because in Chinese men and older Italians, a lot of heavy smokers. And smoking lung damage seems to make you a lot more vulnerable to this. So we don't know exactly, but the 80-15-5 is fairly consistent so far. 80% of people being, mildly ill, 15% of people being seriously ill, 5% of people being very seriously ill, requiring intensive care. Now, here's the problem in Ireland, just for an example. Let's say 1% of the Irish population got COVID-19 at the same time, so 1% were sick simultaneously. We'll say there's 5 million people in Ireland, there's actually 4.92, but let's say there's 5 million people for easy maths. That would mean that 50,000 people would be sick. Now, at the end of the month, projections are 15,000 people, 15,000 people will be ill. 80% of 50,000 would be 40,000. 40,000 people at home, sick, maybe requiring a little bit of medical care, probably not. 15% of people would be 7,500. That would be 11, and in Ireland there are about 11,200 beds. Now, those beds are not all empty. Those beds already have sick people in them. But let's say we have another 7,500 people coming in. Immediately the health service is overrun. And then the biggest problem is 5% of them would be 2,500. Those are people who need ventilators, who need possibly ECMO, who will need urgent care. We have about 350 intensive care beds in Ireland. Well, the number varies between 300 and 420 depending on who you ask. But we have about 350 intensive care beds. We have nowhere near the intensive care capacity to handle those. So while the actual mortality rate, if all 5% of those get intensive care, might be, okay, a fifth of them die, even with intensive care, you're looking at a much higher proportion dying just because you run out of ICU care. It is imperative to slow the spread of the disease to get the health service time to cope. That's why we're doing all this stuff about slowing disease. It's to save as many people as we can. Slow down this. This is always going to kill people, but it's about slowing down how many people it kills to avoid it turning into like something truly horrific. Could we not get more ventilators? Maybe. The problem is you don't just need to produce ventilators, you also need trained medical staff to run them. So hence the HSE is apparently recruiting again. Trying to help with that. In the UK, they were asking about people to produce ventilators, and the difficulty is that you need manufacturing capacity to gear up for that. That's tough, and the other problem is you need people to run them. I know Boris Johnson was talking to people about it, and apparently he referred to it as Operation Last Gasp. So I'll roll my eyes at that one. Anyway. What are the countermeasures we can take up for this? Well, the first one is to avoid people. Easier for some people than others. Easier if you're working from home. Easier if you've been asked to quarantine yourself. But avoid people. If around people, maintain distance. The further you are away from somebody, the less likely you are to infect. I mean, there's numbers going around that you need to be 15 minutes away from someone to get 15 minutes with someone to get infected from them. If someone sneezes on you or coughs on you, you can be infected almost immediately. So, I mean, if it gets into your eye or something in your mouth. So the best thing you can do is avoid people as much as you can. Hand washing, like hand sanitizer is great and all. Hand washing is better in a lot of ways because you wash your hands with soapy water. The soap helps break down the lipid covering of the virus. Also, it washes off the virus, gets rid of the problem. You may need to, if you're hand washing a lot, you may need to moisturize your hands as well as your hands are drying out because you're washing all your natural oils off that. One solution is that this isn't recommended if you're working in healthcare or anything, is to brush back, if your hands are feeling very dry, is to brush back your hands through your hair and they'll pick up some of the oil from your hair. And that can basically re-moisturize it a little bit. Not recommended if you're working in a sensitive area, but if your hands are feeling terribly dry and you're out and about, you can do that. Disinfect surfaces that come into contact with humans. So yeah, again, we were talked about how long it lasts on metal or plastic. Lasts substantially less on cardboard. Are you out and about in the shops? Do you need to worry? If you think something's high risk, give it a wipe down with something afterwards. Now, people have been making up homemade hand sanitizer out of isopropyl alcohol and aloe vera gel. If you can get them, great. If not, soap is fine. The hand sanitizer is mostly handy when you're out and about and you can't find a sink. So I made up some with some isopropyl alcohol and some aloe vera gel, and it's just in a little squirt bottle in my car. But other than that, at home, hand wash, soap and water. Other alcohol, like it can be quite tricky to make it up in the right proportion, so you're best off with soap and water. The hand sanitizer production is going to ramp up. I'd say in a few weeks we'll be fine. We'll have plenty of it. The ultimate counter to all of this is the development of resistance through exposure. So if you build antibodies If you've been exposed to a virus, your body will construct antibodies to bind to that virus's antigens and neutralize it. So the antibodies will attach themselves to the antigens on the virus, and they can neutralize it in a couple of different ways. You can get both a cell-mediated response, where the antibodies bind to the cell that's full of the virus, and a cytotoxic T cell, a white blood cell, comes along, reaches down into the virus cell and activates. There's a cell structure inside the inside your cell called the lysosome, which is full of digestive enzymes, sometimes referred to as a suicide sac. Twists it, that tears open, the enzymes destroy everything inside that cell. That kills the virus factory. The other thing it can do is a humoral response, where the antibodies coat the virus, and your neutrophils, another type of white blood cell, rock up, suck in the viruses, and just break them down with enzymes that way. So that's two ways it helps. To have this response available, you need to have been exposed to the virus and develop antibodies to bind to it, the antigens. Do they work forever? It works until the virus antigens change shape, and that happens periodically through the process of antigenic drift and antigenic shift, where the virus mutates and the shape of its antigens change. That's the big risk on this, that we could become immune to one type of SARS-CoV-2, and then a second type comes along, and then our immunity is gone. Similarly, a vaccine will operate on similar principles, giving people access usually to the antigens of the virus and get their immune system to develop antibodies to it that way, by exposing just to the antigens, not the whole virus. This is the theory behind using herd immunity to fight this, where 80% of the population get the disease, become immune to it, or die, and then the people who are more vulnerable are protected because there are fewer hosts around for the virus. I am skeptical about that as a mainline tactic. It works eventually. If we get a vaccine, the vaccine will most likely work through that method, but a vaccine is probably 12 to 18 months away, just because it takes time to isolate the antigens. It takes time to test the safety of giving people the drug, which is the problem. You could probably knock up something in a couple of weeks, but you wouldn't know it was safe, and you can't be giving people stuff that'll definitely kill them. So yeah, what I will probably do is, in my next microbiology online class, I will go through vaccines a little bit, and then we'll move back onto the more comfortable ground of bacteria and fungi. So I'll probably talk a little bit of vaccines in the next class. Do we have any questions from the chat? And in the meantime, yeah. Is there any research rebalancing the risks between stringent social actions versus potential harms of economic disruption? That's the kind of cross platform or cross skill set learning that will be important. Austerity costs lives too. Absolutely. The difficulty is going through a less disruptive approach is very much it's not, we would, like, if you go, okay, we're going to do this through a less economically disruptive way, we're just going to assume normal operations and we're going to let the people who are going to die. You're talking about letting probably, let's say everyone got it, let's say most, that 5% that get seriously ill die. So 60% of your population get it, or 80% of your population have 5% of that population die, primarily old people, admittedly, but also people with compromised health or compromised immune systems or some other underlying condition. Maybe people are obese, maybe people are thinking, is it okay to kind of directly or deliberately go with a policy of, okay, 5% of this population die, maybe 3% overall the population are going to die, and that's our approach. I'm not sure how politically viable that is. Economically viable. Economics is in some ways mostly made up. If you look at the stock markets crashing at the moment, I mean, you know, they go up, they go down based on kind of investor confidence in a lot of ways. So you know, I'm skeptical about it. I'm skeptical about how easily you can compare both of them to each other. In generational terms, does it benefit boomers and cost millennials? Yeah, to an extent. But, you know, older people deserve to be alive too. Is it long term going to be very disruptive? It is, but what could happen going forward is you might come understand that actually we need a better distribution of income. Like, let's say this goes on, we go through this level of lockdown for 3 months, maybe 6 months, which is might be what is required. So many people will make no money during that time because they've got, you know, very gig based jobs, or, you know, kind of very seasonal jobs, and that season isn't going to happen. We'll have to look a lot about something that looks a bit like maybe universal basic income in some cases, which is a fascinating thing that might happen, that we might have to go, actually, well, we're just going to have to give people money to stay alive. And that money's still going to circulate around the economy because people are going to want to buy food. You know, food and other essentials. So it'll be interesting to see what happens. This is very much kind of uncharted territory. We haven't had a major deliberate economic shutdown like this before. It'll be interesting to see what happens. I don't know if it's going to be bad or good long term. It's kind of outside my wheelhouse in some ways. But I can tell you the science behind it. I can't tell you the economics behind it as well. Alrighty. Well, main risk is crisis come via debt. Crisis collapse, civil unrest to give money. I get what you're going for, but it's beyond my wheelhouse. You know that. Yeah, I mean, definitely. There's always going to be issues with this. You know, I'm not going to cover the full details here, unfortunately. All right, so. For all the students listening, it is quite likely something around that topic is going to come up with the exams we're going to be doing. We'll be looking at definitely one question on viruses between the biology and microbiology papers. Definitely one. Maybe more than that. I'll be doing some more stuff about vaccines in the next class and cover some more, you know, bits and pieces with regards to this. Revisit this as new stuff comes up, but it'll be moving on after that to doing a little bit about fungi and bacteria, and that will help prepare you for the microbiology papers. For my maths students, I will be doing a maths class at some point on this, and then a little bit more anatomy and physiology, probably moving on to the muscles, the skin, and maybe onto the cardiovascular system after that. Thank you all for hanging out. I will see you all tomorrow.